So now we're on 10.2 and 10.3 in this video. We're talking about the formation of proteins. So we're building our proteins from amino acids. So these big polymers of amino acids, and they can be big. They're going to be more than 50 amino acids if they're a protein. And we can also have amino acids that are, just few in a chain, which can be known as a peptide. So we know from the last section that we're going to have a carboxylate ion on one amino acid molecule, and you're also going to have that protonated amine. So you're going to have a carboxylate ion when you're building these peptides reacting with a protonated amine of another amino acid, right? So you're going to build these chains that way, and you're going to have something called a peptide bond between within those structures that you're building. So in this case, whenever you are putting these amino acids together, a water molecule ends up being removed. And you're going to have this amide functional group being formed. So you'll see what I'm talking about maybe a little better on this slide. So what you're going to have is sort of this condensation reaction. All right. So wherever you have functional groups are reacting in these condensation reactions, all right, you're going to have, remember, we've done this before, hydrogens and hydroxyls that can be removed, and you're going to end up having a different functional group that's going to be formed when this happens, and water, although that's not really our main focus, right? You know what a condensation reaction is, and you know that you form water. So if you look here, like you'll have a carboxylic acid, reacting with an alcohol, and so you can see which atoms are going to form the water. All right, so you've got that, and you see how the alcohol ends up being married to the rest of what was the carboxylic acid, and you formed something called an ester. Remember, we've talked about esters already. So you remember these general principles. And you can even see another example here with the carboxylate and a protonated amine reacting together, forming an amide, and of course water. So these are biological condensation reactions that happen to build up these proteins, all right, to put these amino acids together. So if you have, all right, here's another example, all right, so this hemiacetal getting together with the alcohol, forming a glycoside, and still the byproduct of the water. So you see that you have, remember, glycosidic bonds between monosaccharides, right? So you remember kind of doing these sorts of reactions before, all right? So like you can see here, what we're having here, like when you have a peptide bond forming between amino acids, all right? So remember the groups that you're going to have on amino acids, all right, whenever you're studying these examples. So this is like whenever you have peptide bond formation between amino acids to put these big structures together, all right? This is where it's got to start. So you have amides and peptide bonds. So when amino acids are going to combine, the amide bond that is going to occur between two amino acids is called a peptide bond, right? So look, we've got two amino acids here, alanine and valine, all right? You put them together and you see what happens. All right, it's condensation, you formed water. Okay, so you see what comes off of this protonated amine, these two hydrogens, all right, with oxygen. So that forms water. And you see, all right, on your end of where you had your C terminus, all right, it is going to be connected to the N terminus of the valine, all right, that the other end of the valine. So what you have now, all right, is a peptide bond, all right, between what used to be two separate and non-bound amino acids, right? But now you have alanine dash valine, all right? So you have a bond between these two amino acids, a peptide bond, and you can abbreviate that AV like the two one-letter abbreviations too. Okay. So what you can have, the smallest peptide, is just two amino acids together. So if you have a dipeptide, so the N-terminus, the amino terminus it's also called, has an unreacted alpha amino group, right? Remember we talked about the alpha carbon and the alpha amino. So the alpha amino group is going to be unreacted, right? Just open. And then the C-terminus, all right? So that's whenever you're going to have the carboxy terminus, that's an unreacted carboxylate group, all right? So here you're going to have the N-terminus, all right? And here you're going to have the C-terminus, all right? So remember earlier I said to pay attention because you were going to be seeing that later on, all right? So that's really considered to be N-terminus and C-terminus when you at least have two amino acids together. So normally peptides are going to be written out with their abbreviations ordered from the N-terminus to the C-terminus. And you are going to be able to identify all of these parts of peptides in this class for your test. So you can follow these steps to be able to do that. So the first thing you're going to do is circle the amino acid at the N-terminus and give it its name. Okay, so you need to look at the backbone, look to see where that amino acid must start and end based on the parts of an amino acid that you know. And then you're going to put a square around the amino acid at the C-terminus, so the last amino acid, all right, going from left to right, and give it its name. Then you're going to locate that peptide bond by just drawing an arrow to it or putting an asterisk, whatever the direction tells you. So you're going to have to find that peptide bond, right? You remember, you know what it looks like, okay, right here. So then you're going to give the abbreviation names for those dipeptides. So that's classically what you're going to do in the examples for this class. So dipeptides are going to be where you have two amino acids and you form a peptide bond between them. And the first amino acid that has the unreacted N-terminus, that's what's going to come first, all right? It's going to be on the left. And on the right is where you're going to have your C-terminus, and that's going to be your unreacted carboxylate. So two peptides. So if you have, so each pair, all right, can combine to form two totally different dipeptides. So two dipeptides are going to be structural isomers, and they're going to have different properties. So the order of those amino acids matters in respect to other amino acids present or nearby in interactions that they may have with them. So the order of those amino acids is what dictates the structure, and the structure entirely matters for the function. The structure makes the function. If the structure changes, the function changes. Remember that. So structure is very important for function, and structure is all about the order of amino acids because that's going to affect how things fold, how those functional groups interact with each other, because you know that there are some major differences between those functional groups, being polarity, if they're, you know, acid or base, for example, acidic or basic, for example, or neutral. So protein formation again, if you have a compound, all right, having amino acids that are joined by a peptide bond. Those are all going to be called peptides. And you know, the smallest ones I said are dipeptides. All right. So when you've got 2 amino acids, if you've got 3, then you've got a tripeptide, 4, a tetrapeptide. So you know some of these prefixes already. But with the more amino acids, you're eventually going to call this a polypeptide. If it's, you know, you have 14 amino acids, maybe you just still call it, people will still say a peptide. But when you get more and more amino acids, you refer to this as a polypeptide. And if you have a biologically active polypeptide, meaning that it has some sort of function in the body, and it has typically 50 or more, sometimes a lot more amino acids, that's going to be a protein. It can be thousands of amino acids, all right? That's going to be a protein. So let's talk about some peptides in an applicable setting. So you'll have some patients that might have celiac disease, and that's a pretty rare disease, relatively speaking. But what causes celiac disease? And this is where peptides come into play. So something that you've probably seen at the store is, you know, a package that says gluten-free. Well, what's gluten? So that's a mixture of proteins that we find in grains, and something that's a grain that we eat a lot is wheat. So you've got gluten there. And some people, you know, will buy these gluten-free products because they might have celiacs. They can't have gluten. And that's because gluten has 2 main constituents, glutenin and gliadin. And those 2 main proteins in gluten have areas that are rich in proline and glutamine. And that's sort of a unique sequence, all right, a unique little bundle of amino acids. So when gluten gets into your GI, once that's consumed, some of those peptide bonds are going to be broken up by enzymes, proteases in particular, because we're talking about, so proteins, all right, proteases, that they break down gluten. They're going to leave behind a bunch of proline and glutamine, all right, peptides. And so some of the glutamines are then going to be converted by an enzyme called glutaminase to glutamate, all right? And so when this happens, the side chains in the glutamine hang on a second, I have to fix this slide. Ugh. Okay, so when this happens, whenever the glutamines are modified to glutamate, basically the side chains are going to change, right? So whenever you have that change, your body is going to say, whenever it recognizes glutamate, you're going to have special senses in your body that are really specific, and that are going to say, well, you know, we don't recognize this so much. So whenever you're changing the transition, so whenever you have this transition and you're going from a polar neutral residue to a polar acidic residue, this is not ordinary. So this is, you know, outside of homeostasis, right? You're going to have a negative charge on those peptide sequence, and your body is going to form an immune response against that, against those peptides. And so this causes a lot of discomfort in the gastrointestinal tract. And these people with celiacs have to avoid gluten. You know, something that can happen from this could even be malnutrition. You have to be, you know, really careful about avoiding gluten if you have celiac disease, because you don't want this buildup of, you know, unfamiliar amino acids, right? It's a pretty rare disease. I have a friend who has it. It's actually a really terrible disease. So let's talk a little bit about the 3D structure of proteins. I think this last section rolls right into this one pretty well, and then we'll work some problems. So we are stringing together these amino acids. So we talked about just going from an amino acid. Now we have a dipeptide, we've got this N-terminus and C-terminus on the ends, and we know that we can have a polypeptide and we can have proteins. So we have a primary structure. A primary structure of a protein is the order of those amino acids. That's just the order of the amino acids that are strung together, which we know is important. So in this case, the amino acids are just joined together and they form a protein backbone. Okay, so this is the backbone that you see there, pretty much all circled in orange. So what's coming off of that backbone are the side chains, those R groups of those different amino acids. Okay, and that's going to affect the structure later on. But we're just talking about the primary structure. This is just the primary structure. And those side chains are going to be dangling off, okay, to the side, all right, making this a unique structure, okay, those different side chains. When we have a secondary structure, all right, here we're going to have a protein, all right, that might have some coils, right, and it might have some sheets. Okay, so you're going to have coils or other patterns within a three-dimensional shape of a protein. And those coils and sheets are secondary structures. So we call the coils, like a coil on a phone, an older phone, an alpha helix, and a beta pleated sheet is kind of like, you know, if you think of like a folded sheet, like there's one big pleat, then it folds over, then there's another big sheet right next to it, then it folds over, there's another big sheet right next to it. And you'll be able to see that on the next slides. The secondary structures are stabilized by hydrogen attractions, or we call it hydrogen bonding, although it's not even a real bond, right, along the protein backbone. And it involves the amino acids that are close to each other, all right, that are really close to each other. So just remember the hydrogen bonding that I said there is just an attraction, right? So those held together, those secondary structures by those hydrogen attractions. So that alpha helix, right, is a coil, right, like the phone coil, like you see here. And it's stabilized by the hydrogen bonding or attraction between the carbonyl of one amino acid and the hydrogen atom of another amino acid. So you remember your polarity and your partial positives and negatives, right? Depending on electron distribution. So it's going to be the carbonyl of one amino acid and the hydrogen atom of an amino acid on the fourth amino acid away from it, all right? Because that's whenever they're going to come close to each other in this coiled structure, if you think about it. So that's where the attractions lie, okay? And that's how this coil is formed. So the positioning of those bonds is what allows the helix to stretch and recoil, okay? So the side chains are what's going to be projected outward. The side chains are coming off. They're projecting outward. They're not going into that core. Those side chains are going out, away from the axis of the helix. They're going out. So, and you think of that because that's going to have a lot to do with the interactions that happen because of those secondary structures. Like a beta pleated sheet, okay? So that's where you're going to have like, well, I may not be the best artist, but you're going to have an extensive structure where the segments of a protein are going to kind of line up and form a zigzag, like a paper fan. And so sort of more like that. So these strands are going to stick together side by side, attracted to each other through the hydrogen bonding or attractions between the backbones. So here you're going to have those side chains going above and below the sheet. So still projecting outward, okay, above and below the sheets. So there's significance to understanding these secondary structures, because we have to know the optimal way of things to understand when a problem occurs and why. What happens with Alzheimer's disease? Well, that happens whenever something is not right with one of these secondary structures. So you've heard of Alzheimer's. A lot of times it happens in people past the age of 65 where they can't remember things anymore. It's a really, sad and terrible disease. And it can interfere with what these individuals need to do on a daily basis. And they have a distinct, a distinctly defined problem with their nerve cells. So what's happening is, if you look at the morphology of the brain tissue of these people that have Alzheimer's, they have plaques between their nerve cells. They have problems outside of and inside of the nerve cells that ultimately cause the nerve cells to die, and they cause the terrible symptoms that you see. So what happens is, you're going to have, I think we talk about it on our next slide a little bit, where they're going to have a problem with their secondary structures. Where they should have alpha helixes, they're going to have beta pleated sheets. And you're going to have a buildup of these plaques because of the secondary structures between the neurons. And then you're going to have neurofibrillary tangles within the neurons. So outside and inside the neurons, you're going to have problems, and you're not going to be able to have information transfer between the cells, which you need between nerve cells. And so without communication, your nerve cells will die. So specifically, this slide talks about what happens with those secondary structures. So those plaques are going to be made of a protein that's called beta amyloid protein. Beta amyloid protein. So what happens is, when you have somebody who does not have Alzheimer's, the beta amyloid is alpha helical. So without Alzheimer's, the secondary structure there is going to be alpha helix. But in a person with Alzheimer's, right, the beta amyloid protein is going to have beta strands. It's going to be built up of beta strands. And you're going to have a secondary structure that is super sticky in these folds, and it's going to form these aggregates that you wouldn't have had with the alpha helix secondary structure. This is what forms the plaques. And there is no cure for this. There are only medications that can slow the progression and reduce the symptoms for a time. There are people that are working on vaccines targeting beta amyloid and those particular secondary structures that are outside of the norm or what's optimal, not diseased. So let's move on. But you can see that this is what you would have without the disease, and this is what happens with the disease. So you can see here that there are these plaques that build up on the outside between all of these nerve cells. All right, you see the nerve cells, and you have these neurofibrillary tangles within the cells too. So you really have problems with cell-to-cell communication. And beyond the secondary structures, we talked about primary and secondary. Now we have tertiary structures. So this is where you have alpha helices and beta pleated sheets of a polypeptide chains interacting with each other in the environment. right? So it might be in an aqueous environment. So those nonpolar functional groups, those nonpolar side chains are going to get away from the water and they're going to go inside of a protein, the interior, where the polar side chains are saying, hey, how you going? How you doing, water? And they're going to be on the outside of the surface. So you see how those different functional groups affect the entire fold of a protein. So the tertiary structure is therefore stabilized by attractive forces between those side chains in the environment, away and towards the environment. It's all based on these side chains. So that's how we're going to get the three-dimensional shape. And you can see here, and we'll talk about each one of these. We're going to have hydrogen bonding with water, all right? You have hydrophobic interactions where you have like those London dispersion forces we went over. Some amino acids are going to be able to form disulfide bonds with similar functional groups to nearby amino acids. You're going to have hydrogen bonding interactions and something called a salt bridge. So let's talk a little bit about these. So you're going to have those nonpolar interactions where amino acids are going to repel away from the water and aggregate on the inside of the protein. And then you're going to have polar interactions. If it's polar, the amino acid side chains are going to want to embrace water, right? You're going to have all that hydrogen bonding and dipole, ion dipole interactions, depending on the functional groups. And then you're going to have something called a salt bridge. You probably saw that in the previous slide. So that's whenever you're going to have the acidic and basic amino acid functional groups, the side chains, that are going to be in ionized form in an aqueous environment, depending on that environment. So those opposite charges attract, and you're going to have a really stable ionic interaction, and that's the salt bridge. Okay. And we talked about a little bit about the disulfide bonds. So you might have two amino acids with thiols that are nearby, and through an oxidation reaction, you can form a disulfide bond, okay, with those two sulfurs. So you can see there, that's all oxidation and reduction. So it's the principle of whenever you are getting a perm, you can use different chemicals that can, cause the breakage of disulfide bond. Like you can try and get a perm in your hair. So what you really want to do is you want to break the disulfide bonds that are in like keratin, like in your hair, those fibrous proteins. You want to break up that disulfide bond so that you can loosen those interactions. All right. And so once you loosen these interactions, or hang on just a second. All right. So you wanted to reduce. So you have this oxidation reduction reaction basically whenever you're trying to curl your hair, for example, where you can reduce these bonds and, reshape your hair around a curler, for example, and then allow these bonds to reform again. Okay. So whenever we're talking about the three-dimensional structure of proteins, and we're talking about the tertiary structure, we know that we can have both globular proteins formed and fibrous proteins. So globular proteins are going to have this compact spherical overall shape with the polar amino acid side chains on the surface and the non-polar on the inside in the core. And so this is what's going to be true of many enzymes and cellular proteins. When you have like your skin and your hair, these are fibrous proteins, all right, like keratin. They're thread-like structures, very strong, very durable. So note that those are some differences, like you have keratin and collagen in your hair and skin, respectively. So beyond the tertiary structure, you're going to have something called a quaternary structure. So we had our primary, which was the order of amino acids, our secondary with our alpha helix and beta pleated sheets, then our tertiary structure where we had interaction between those secondary structures, and we can have quaternary structure, all right? So those are going to be whenever we have interactions between 2 or more polypeptide chains to make one single biologically active protein. That means something that functions. So the individual polypeptide chains or subunits are going to be held together by the same interactions that stabilize the tertiary structure. So in some cases, we need to put together these big polypeptide chains to have something that functions. But we don't always have a biologically active protein with a quaternary structure. It's not always necessary. Sometimes we just have one polypeptide chain. So here's a table that sort of organizes all of that information, where you have primary structure, those are peptide bonds, secondary hydrogen bonding attraction, all right? And tertiary, you're going to have all of these different types of attractions. It depends on what functional groups are present, really. And your quaternary structure is going to be a lot like tertiary, except you're going to have these big subunits put together. Okay, and so just a picture of this here, and you see this big globular quaternary structure here, right? Remember, this is your alpha helix secondary structure, so on and so forth. So you can kind of see the differences in primary, secondary, tertiary, and quaternary. All right, let's go over some problems, and then we're going to be done. Nope, those aren't the ones. These are the ones. All right, so you've got this table here for common condensation reactions between functional groups. Okay, so let's do, we're doing the odd ones here. So let's do 10.13. Okay, so what are the products for the following condensation and hydrolysis reaction? Well, for A, we know that we have the addition of 2 amino acids, right? So this is going to be condensation. If we look at B, all right, here we have water. So it looks like, are we going to have condensation or hydrolysis, right? So you tell me that. So here we're having a condensation reaction, right? Here we're having a condensation. Here we're going to have hydrolysis. So if we have condensation, we're going to be putting these 2 amino acids together, all right? So how are we going to do this? Well, you know you're going to be making water, but what's important are those 2 amino acids joining together to make this dipeptide, right? So that means we're going to be making this peptide bond. So let's draw our structures here. So we're going to have, let me move this water out of the way. So we're going to have our structures. All right, we have our hydrogen, we have our functional side chain, we have our other. So I've drawn most of one of these amino acids. And so what's going to happen is 2 of these hydrogens and this oxygen are going to get together and make a water. So then we know that right there, we need to make a peptide bond between this carbon and the nitrogen of the next amino acid. All right, and we're going left to right. So that's how those problems are going to work. So we're going to have nitrogen and our hydrogen, and we're going to have the rest of our amino acid there. Finish up this C-terminus there. And there you go. That's how you do. So just make note of where you make that peptide bond and understand what type of reaction you're doing, and it'll never fail. Okay. So let's see. Oh, we'll have to do this hydrolysis. Okay, so basically this water is going to come and break up what we've got here. So what we have, what we're going to end up having, all right, is right, and you can write it a few different ways, but we're going to write it the way it is in this problem. That's exactly what's going to happen. All right. So there you go. And we've done hydrolysis before. It's just like that. Okay. So let's see. Let's do, are we going to do 10.15? Yes, we can do that. So 10.15. So consider the following tripeptide. Okay, circle the N-terminal amino acid and give its name. Okay, well, if we look here, so we can find the beginning and the end of an amino acid, right? We'll know that this is glutamine. Glutamine, that's GLN. Glutamine, cutamine, remember? Oh, it kind of helped. All right. So then we have histidine. Okay, so we have our next amino acid, all right after peptide bond. And that's going to be histidine. All right, so look at some of the things that are unique about that. Make yourself a table. And in our C-terminus, this is our N-terminus on this side. And we've got our C-terminus. All right, that's going to be threonine. And that's THR or T. Okay. All right. So that's the C-terminal amino acid is threonine. All right. And we've done everything. Okay. So 10.17. I think we can do those in class. All right. Did we do just about everything? Let's see. I think we can do 10.17. Let's do A. Okay, and let's do it right here. Okay, draw the structural formula for each of the following peptides. So let's see. What do we need? What do we need here? All right. So we have, all right, let's see, phenylalanine. Let's draw this out. So glutamine. So if we have, draw our backbones. Okay. And we know that we're going to lose an oxygen and two hydrogens in this condensation reaction here. So because we have a dipeptide, right? So we have, this is going to come off, and we have our N, let's just say, oops. We have our protonated amine right here. CCOOH, CH2, CH2, carbon. All right. So what's going to happen are these two hydrogens, all right, and oxygen is going to come off, and this is where we're going to make our dipeptide bond. Okay, so now we're going to have, oops, our N, oops, All right, so we have our N here, and we just have N, and we are going to have our bond to that carbon. All right, and that's it. Oh, I think originally I forgot to draw this here. Okay, yes, so, all right, so that looks right to me. So you have your N term and your C term, and there you go. That's all you have to do for those. And similar if you have 3 amino acids, right? And so we'll work some of those in class. But I think this is a good place for you to get started. All right. So are we going to do some 10.3 problems? Yes, we are. Let's do 10.3. All right. So let's go over 10.21. Let's not do red. Red's kind of scary. So what type of bonding is present in the primary structure of a protein? Okay, well, in the primary structure, right? Remember primary, that's at the beginning. That's just the sequence of amino acids. So those are just covalent bonds, peptide bonds. All right, so you're still going to have that and you have your primary structure, but that's just the sequence of amino acids. Okay, so that's it. We're not talking about folding or anything like that at that point. So 10.23, name the stabilizing attractive force in secondary structures of proteins. So those are like the alpha helixes and the beta pleated sheets. The beta sheets. So you're going to have hydrogen bonds. It's like that attraction, remember? So those are the backbone interactions. Those are going to be backbone interactions. Right? Because we're talking about secondary and not tertiary. All right. So let's see. Let's do 10.25. Describe the differences in the shape of alpha helix and beta pleated sheet. All right. So we've got an alpha helix. All right. We've got something that is coiled. We have amino acid side chains that are pointing outward. All right. And we have hydrogen bonding along the axis. Every 4 amino acids. Okay, and so remember that's something like, and the R groups are on the outside. Okay, and then we have a beta pleated sheet where, oops, I don't know why I put 2 T's on a sheet. And you're gonna have, just writing too fast, I guess. All right, think of like this fan structure where you're gonna have some attractions. All right, so here you have sort of, you can call this an open extended zigzag. It's open, it's extended, it's zigzagging, and you have side chains that are going to go above and below. Side chains, so that's the R groups, extend above and below the sheets. Okay, so in both cases, you're going to have hydrogen bonding in the backbone. All right, so I think that's good, and we can go over the rest of this in class. All right, have a good day.